Welcome back to another episode of Bioconversations Demystifying Science. We've been exploring the world of analytical sciences and just covered the critical role of physicochemical characterization, where we analyze the structure, stability, and purity of biologics. Now, it's time to shift the focus to another essential pillar of analytical sciences, which is the functional characterization science. Because once we know a biologic is structurally sound, we still need to ask the question, will it work the way it is supposed to inside the body? That's where bioassays come in. And that's the journey we will be exploring in today's segment. To guide us through this, I'm thrilled to welcome Malini Subbarao, Senior Scientific Manager and an expert in bioassay development at Biocon Biologics R&D. Malini comes with a broad experience of 17 years and she has been developing and validating assays that measure how biologics affects the body and brings about the change for which the drug is prescribed. Malini, welcome to Bioconversations.
Pleasure to be here, Kavita.
Before we segue into the actual topic of discussion today, which is the functional science, let's get to know you a bit better. So Malini, you've been with Biocon for over 17 years. In these 17 years, what has been your greatest learning experience?
I think these 17 years has been where the organization grew and I grew along with the organization. And one of the biggest learning has been that growth comes disguised as challenges.
And opportunities as well. So, and we don't have a shortage of either.
Yes.
Problems is what helps us innovate.
True.
So, you have evolved from working in the lab on individual assay development and now you guide a team of scientists who develop these assays and you are basically strategizing this. What part of this transition has been greatly rewarding to you?
I always loved working on the bench. So that's something I miss doing it. But now with a bigger perspective of what is the expectation from the regulatory agencies, today it is easier for me with the experience that I've had to tell people what is required, what is the expectation, so that they can work on in a way that it meets all the requirements from the agency.
So now that we are into this topic, could you help us understand what are bioassays and why are they important?
See, bioassays are basically tests that show that the drug that we take in does the function or the action that it is supposed to do in our body. For example, if we take insulin, it's taken to bring down the glucose level. So we do a bioassay development which will demonstrate that glucose is being taken up by the cells. So that's the kind of analogy I can give as to what bioassays do.
So you have a target for which you are testing it against?
Yes, there is a specific target to which the drug would bind and there would be a response.
So insulin being one, so different drugs act on different targets, which means so when we say a target, it is essentially a receptor.
It may be a receptor or it may be a soluble protein. It depends on what is the drug that we are looking at and what is the response that would vary on it.
So when you are developing these bioassays, what are the challenges that you encounter?
Bioassays are done mostly in an in vitro environment where you have a
In vitro means?
In vitro means outside the body in a culture area which is completely controlled format.
So that is basically a surrogate.
It's a surrogate to the what is happening. In vitro is more of it is inside a Petri plate or a flask if you want to draw an analogy.
Right. So, but you would be culturing something like a cell. So those cells will be representative of the receptor or the target.
The cells that we select, they would be specific to the target that we are looking at. If we are looking at a oncology presentation.
Such as a map.
Such as a map. Our trastuzumab biosimilar that we do have, it's targeted against breast cancer. We take breast cancer cells and then test the drug on those cancer cells. Okay. So it's a very specific environment in which we do the testing.
So when you are testing this, what are the outcomes that you're looking for? Or the effect?
So there are different kinds of effect each drug has. For example, if you look at insulin, it will not just act on glucose, it also affects fat metabolism. It affects different functions in the body. So we will look at other binding aspect of insulin to its receptor, the adipocyte function that it
Adipose,
activates,
which is responsible
for the fat metabolism. And say, for example, we look at MAPS. It can be of different kinds. It will be binding again. It can be receptor phosphorylations, or it can be cytotoxicity.
So what is cytotoxicity?
Cytotoxicity, you can split it as cyto means cell, toxicity is harmful. Or we look at, and when we usually talk of cytotoxicity, we look at cell death.
Right.
So it can be death in different forms. One would be cytotoxicity or apoptosis or proliferation, inhibition of that proliferation.
So basically, if to put it simply, you have an anti-cancer drug. So that is expected to kill the cells.
Yes.
So it would be killing not just this cell, but there would be other actions also which may result in unwanted toxicity. Is that what you also look for?
Yes. The toxicity part with cancer is more specific because it's always binding to the target and then activating the response. But say in case of insulin, which is a chronic dosage, insulin can also bind to insulin-like growth factor receptor.
Right.
So that can cause a growth of unwanted growth in different regions of the body. Undesirable effects. So we test even for those that say the product that we make in insulin is similar to what is produced in our body. The extent to which, say, it will bind to the growth factor is similar to that. So when we are able to show that, it means that the side effects are similar to what endogenously produced insulin would be, not very different.
Understood.
We also see how the cytokines production happens. And
so cytokines is essentially, again, cell is cyto and kines is
messengers. So these are signaling proteins which will act on the cells, and they can activate the cells or they can stop an action. So basically these are messenger proteins which are secreted by...
And you measure those. Basically,
we also measure those.
You want to see whether it's going up or going down for a particular indication.
Yes, right. We do see that.
So how do you measure the cytokine release?
It can be through ELISAs or any other...
ELISA is an abbreviation, I suppose.
Yes.
It is...
ELISA is nothing but enzyme-linked immunosorbate assay.
Okay, that's a very big term. In short, what does it do?
So basically, it looks at how the target binds to the drug, and we measure that response chemically.
Oh, so that's where you use those 96 well played, which we keep seeing in all our science books, maybe.
Yes.
Okay. Yes. So, ELISA as a standard, is that a standard test that you do?
Yes.
For regardless of any molecule?
For most of them. For most of the monoclonal antibodies, we do ELISA.
So you do a receptor binding, you do an ELISA, and you do cytotoxicity, apoptosis, proliferation, inhibition. So there are a whole battery of tests.
Also do binding kinetics in addition to that.
And how does that help?
So it tells us what is the strength of binding. Usually when we do an ELISA, it tells it is binding, yes or no kind of a response more so.
So you want a yes or a no? I mean, it should bind. Technically, the drug
binds. And we see like it's a static reaction that we are seeing. But in the body, it is never a fixed reaction, right? As the drug increases, the binding to its target would also increase. So the dynamic increase in binding and change in the system that it would happen because of this binding, that affinity is what we will determine. So when you have a drug and a target, it's a basic chemical system, right? It can interact.
It's like a lock and key.
It's a lock and key. So it will interact, but there are a lot of targets and a lot of drug molecules as well. So each of them bind with a different strength. So that strength is what measured in the SPR or the kinetic assays that I was talking about.
I think this is fairly complex, but regardless, it is a concept that is quite
well
established. Yeah. So what are when you're developing all these tests, let's call them assays or tests interchangeably, what are the challenges do you face?
See, one main challenge that we see, especially with these cell-based assays, is the cells itself.
The cells by itself.
The cells by itself. These are living organisms, if I have to say. These are living entities. So the way I am not the same today and tomorrow, or any person for that matter, the cells also have their own behavior.
Depending on the day, you mean?
Depending on the day, depending on the hour, how well fed we are, how underfed we are. So the same exact things also happen with the cells. How well fed they are, how underfed they are, their responses change based on those aspects as well.
That's a very interesting analogy you make. So then what do you do to ensure that they behave same or similar on an everyday basis, because your tests need to be standardized?
One thing that we do is we have elaborate procedures which details every step that has to be followed, and that has to be followed religiously.
Which is called as
the SOPs, the standard operation procedures, are written in detail. And we also use multiple reagents in the assays that we do. So each of them are qualified. So qualification is telling that a product is fit enough for the purpose that we are using it. So we qualify the reagents, we also qualify the methods. So all the whole system is qualified.
So basically, to ensure that a particular reagent, a particular test, and the condition is harmonized or made consistent across. So no matter who does it tomorrow, if I do the test or you do the test, it behaves similarly.
Yes.
Okay. So that requires a lot of precision.
Yes. So the development part is where the all these things are set in. If the development is done well, then you will see less challenges later on.
So I know you mentioned about using an SOP or a standard operating procedure to harmonize these things and to list down to as much detail as you want to. But this development primarily happens in an R&D setting, in an R&D lab. But your products are released from manufacturing and from different sites. So how do you ensure that the communication is, clear, and they also give you the same results that you have developed? So how do you ensure that robustness?
So what we do is, once the method is developed, we qualify the methods as well, like I was mentioning earlier. So these qualified methods are then transferred to QC, and as a part of transfer, we train the QC analysts also
on the nitty-gritties.
On the nitty-gritties of which points they need to be more careful about. These are because they are also going to handle cells which will be again behaving differently if you don't handle it the same way. So we give them the details of how the cells have to be handled, and we also train them on the procedure that needs to be followed in. And once they are familiarized with how to do the things, they execute it in their labs and validate it in their site as well. Qualification and validation are mostly similar procedures, but they are carried out of different sites. Because we are an R&D facility, we call it as qualification of the system, that it is fit for the purpose that it is intended to. In QC, which is the manufacturing environment, which is under a GMP kind of a control, there you validate that yes, indeed, the results come the way it is expected to be. So that's the only difference, at least in bioassays, between qualification and validation.
Right. So what are the different cell lines that you use for studying these bioassays? Case in point being, when you are growing the cells, as we discussed in our previous segments, that mammalian cells or your pichia or your yeast or microorganisms from which we make our therapeutic proteins, like CHO cells, for example. So are these the same cells that you use for studying bioassays, or the cell lines different?
So these are different cell lines. Mostly what you were mentioning is used for manufacturing because they're good at producing the product more. But what we are looking at is not a production of the product at all. It is more in terms of the target. They may be primary cells, like the way I was mentioning, cancer cells, or some of them are engineered cells as well, wherein the response to a particular target is provided through a gene modification, which will give you a luminescence kind of a signal. So it's easier to read out rather than performing a very elaborate assay. This will give us a response within a day or two.
Because you want to also ensure that the tests are such that you get a result in a quickest manner as possible.
Yes. And also it is QC friendly. So most of these reporter assays, which are which have cells which are modulated, we use it for our release assays, so that QC doesn't have to do an elaborate procedure during the product release, but yet we get the same quality of data.
So release test, when you say release test, it means the test that you do before the product is out in the market to ensure its efficacy, safety, or whatever it is you're trying to say.
Yes. So whenever a product goes in, there is a COA which usually goes, which is a certificate of analysis telling this is fit for purpose. So as a part of that tests, we include bioassays as well.
Understood. So when you are tests, what are the challenges that you face when you are sending a particular method or a procedure to the global labs, or from one site to another, are there site specific challenges?
Sometimes it does happen, because within the organization, at least here in Biocon, we have a standard setup. We know what is there in the receiving lab. But sometimes when we transfer it to a different lab in a different region or a country,
the originator lab
and they become the recipient lab.
That's the receiver or a recipient lab, whichever lab it is. Is the lab that receives the procedure that you develop.
Correct.
Okay.
So there the setup might be different, the instruments might be slightly different, the makes would be slightly different. So the output may get affected because of those. So there is an evaluation that we will have to do, telling that whatever they have, the equipments, the instruments are similar to ours, and they can execute it there as well.
And if it's not similar?
Then we recommend them to get a similar product or instrument setting. And
that could be some challenges associated with that, I presume, or it has not been so much because these are standard equipment.
These are standard equipments for bioassay labs. So any bioassay lab we go, they will have these standard set of equipments.
Yeah.
So usually we don't face a challenge, but we do a check just to ensure that the output is consistent. So for that
and then you can measure the difference.
Yes.
So are bioassays only important during the development phase, or is it a test that you routinely do even as the product life cycle management is ongoing?
So bioassays are required from the beginning of the program throughout the life cycle. So as a part of development, we do certain tests. As a part of the product life cycle, before we go to the regulatory agency and get a approval, we do a lot more tests. And as a part of routine release also, there are bioassays. So it's there through and through the life cycle of the molecule.
Are there any automation, futuristic, this is a very futuristic question. Many of these procedures that you mentioned are very routine, very intense. So what are the automations that you think will help in this industry or, in this particular area?
So as of today, there are automations available commercially for performing cell-based assays as well, but they are extremely expensive just to have the setup built in. And also it is difficult for us right now with the extent of programs that we are doing in. We have to build in the analytics also into the softwares, into the tools, so that everything happens outside without human intervention. That's the future.
So again, many people for many segments, in our previous segments also, they have really spoken about artificial intelligence and data science techniques. So do you think this field will also benefit from innovations?
Yes. So analytics will be required at, say, what happens in bioassays. We look at the cells and see whether they are ready for performing the assay. Now, if we want to remove the analyst out of it and bring an automation in that ability to see what is optimal would again require AI/ML.
You'll have to train.
Yes, we have to train AI/ML models to say this is the kind of culture I need to have for performing this particular assay. This is with this cell line it's different, with another cell line it's different. So for each of them we will have to develop that analytics before we completely move into an automation kind of a system.
So before we wrap up, Malini, let's have a rapid fire round where we get to understand your perspectives on few things. You've been in Bangalore you basically all your life. Yeah, I was trying to find out, so I know you for past 14 years since I've been here. So, and you've been a Bangalore girl. How is it that you never explored opportunities outside of Bangalore?
I think there are so many opportunities here in Bangalore that I never felt a need to look out.
Basically, all that you needed to have an enriching life or a career.
Everything was there here. Yes.
And being an accomplished women scientist, what is your advice for Gen Z women, especially in STEM? You've been a STEM senior leader here now, I mean, with time.
Yes. I think one couple of aspects is focus and dedication. And your passion drives your focus and dedication. So if you're passionate about it and really interested in that subject, I think that will give you what success you're looking at.
After so many years developing bioassays, being in R&D and leading the way, how do you stay curious? What fuels your curiosity?
I think curiosity for me comes from trying to understand why things work the way they do. So that question always lies in everything that we do. So that continuously drives my curiosity.
And what is it in R&D that still gives you a sense of thrill or achievement after all these years? What keeps you inspired?
I think it's more of I'm doing what I love doing it. So,
but you miss going on the bench and actually doing
yes.
it though. But you like driving the tests and understanding the regulatory landscape.
Yes, the regulatory landscape, and now I might not be physically doing it, but the design part I'm still contributing quite a bit into it. So that keeps me satisfied that yes, I'm in touch with the bench, though I may not be at the bench.
That's true. You don't have to be. You need to have an understanding of what happens at the bench, largely. And if you do very complex stuff, I mean, all the words and whatever you have explained in this segment, if a fourth grader comes to you and asks, what do you do? How would you explain it to a fourth grader?
So I think it's the simple way to say what I do is we develop tests which will show that a drug works or not. It's basically, say if we use a pen, it's used to write something or draw something. So we're doing something similar. We're using a drug for some disease. Is it curing the disease? Yes or no? Those are the kind of questions. Work that we do.
So you have seen the evolution of biologics over the last decade or so. What's one advancement in science you're most excited about in future?
I think the automation and bringing in the AI ML. I know there's a...
I was just going to say I should have said anything other than AI ML, but no, you are also excited about AI ML.
I think bringing that in decreases a lot of dependency and variability that would be associated with each individual. The way I see something, both of us see red, but it's not the same shade of red that we are talking about.
That's very nice way to put it, yeah.
So those differences are what will get,
essentially eliminating subjectivity.
Yes. So that's what the AI ML and all the automation can bring in. And that's what I'm looking forward to.
Thank you, Malini, for sharing your insights and taking us behind the scenes of bioassay development. It's fascinating to see the science that ensures biologics exactly do what they are intended to do. We really appreciate your time today with us at Bioconversations. And to everyone listening, thanks for joining on this deep dive into functional side of analytical sciences. Until next time, take care and stay curious.